All right. Good afternoon. It's 12 o'clock Central, and it's Wednesday, so that means it's time for the Iowa Learning Farms webinar. Last one of 2022. So the next one, next time you see us, will be 2023. I'm Matt Helmers with Iowa State University in the Department of Ag and Biosystems Engineering, Extension Ag Engineer, and Director of the Iowa Nutrient Research Center, and also a faculty member involved with the Iowa Learning Farms project. And so it's my great pleasure today to introduce our speaker, and we're going to hear about embracing real-world variability to improve cover crop outcomes. Etienne Sutton is a PhD candidate and USDA NEFA predoctoral fellow at the University of Michigan School for Environment and Sustainability. I think it's a great way to end 2022 hearing about cover crops, a really important practice for Iowa and much of the Midwest for helping with soil health and reducing nutrient loss. So, and thinking about the real world variability of that is vitally critical. So with that, I will turn over the virtual floor to Etienne. As usual, if you have questions for her, feel free to type those in the chat box and we will get those to her at the end of her presentation. So with that, the virtual floor is all yours.
All right, wonderful. Thank you for that great introduction. So yeah, as he mentioned, I'm a PhD candidate in Dr. Jennifer Blesch's Soil and Agroecosystems Lab at the University of Michigan. A lot of my research focuses on ecologically based strategies for improving the sustainability and resilience of agricultural ecosystems. And so my talk for today is titled Embracing Real-World Variability to Improve Cover Crop Outcomes, which may sound sort of vague at the moment, but by the end of this talk, you'll hopefully understand what I mean by real-world variability, why it's important for agricultural research, and what we've learned from it so far in the case of cover crops. So as I'm sure many of you know, cover crops have huge potential for improving agricultural sustainability across a wide range of farming systems, with benefits ranging from improving soil health and water quality, providing weed and pest control, supporting pollinators, just to name a few. But one major caveat here is that cover crop growth and benefits can be highly variable across different farming conditions. These two photos are a great example of this, where we have two fields of cereal rye from an on-farm experiment in spring of 2021 that were planted the same way in the same county at the same time, but still had really different outcomes when we sampled them in the spring. And just as a little sneak peek here, in the case of these two fields, it ended up being the soil that was in large part responsible for the differences we saw between the two fields in terms of cover crop performance. So more to come on that later. Beyond soil, there are, of course, a lot of other factors that can influence plant growth, some of which are environmental, like climate and topography, and others are related to management practices, like nutrient management and planting strategies. So taking into consideration all of these different factors affecting plant growth, my research really focuses on figuring out how and why cover crop outcomes vary across farms so that we can begin to identify what works well and what doesn't across different growing conditions, and in turn, help farmers maximize benefits from their cover crops. To do this, I'm using two different approaches as part of my dissertation research. The first is a citizen science study that uses observational data collected in partnership with farmers from across the Great Lakes region to look at how a wide range of factors influence cover crop growth. And the second is an experiment on 6 farms in Michigan that looks specifically at how soil health influences cover crop performance and involves much more detailed measurements of plant and soil properties. Before I dive into the details of those two projects, I want to introduce another common theme throughout this talk, which is understanding how the performance of multi-species cover crop mixtures compares to single-species cover crops. So one of my central hypotheses is that cover crop mixtures may be more beneficial than single-species cover crops, First, because having multiple species growing can increase the chances of at least one species doing well under a certain set of growing conditions. And second, if those mixture species are also functionally diverse, they have potential to support a broader and more robust set of ecosystem services. For instance, the photos that are shown here are of 3 functionally diverse plant types. So legumes like clovers and vetch can provide new nitrogen to farm fields through conversion of atmospheric nitrogen into plant available forms of nitrogen. And then grasses like cereal rye have extensive fibrous root systems that can help scavenge and recycle soil nutrients. And then brassicas with their large taproots can often help reduce soil compaction. So mixtures that combine complementary plant types like these have potential to use resources more effectively, be more productive overall, and support a broader suite of benefits than single species cover crops. So we'll return to these themes throughout the talk, but for now, let's dive into the first of the 2 cover crop projects that I mentioned. In this citizen science project, we're partnering with Great Lakes farmers to gather data on cover crop growth and key factors that can influence cover crop outcomes on real farms across the region. By partnering with farmers to collect this information, we're able to expand the scope of the study to include far more locations and types of farms than would otherwise be possible using typical research approaches. And then in turn, the data that farmer partners report helps inform context-specific recommendations for cover crop management. Our citizen science approach involves 2 main components: a farm and management survey, and then a field assessment. The farm and management survey asks questions about different factors that could influence cover crop growth, like planting strategy, seeding rate, soil type, and management history. And the field assessment is what allows us to quantify cover crop growth across lots of different fields. So during our first season of data collection this past spring, we asked all participants to visit their cover crop field to collect 2 types of data: canopy cover photos and then height measurements. For the canopy cover photos, participants collected photos like the one you see here that capture the cover crop canopy at 3 different locations in the field. And at each location, they also collected several plant height measurements. We're then able to take the canopy cover photos and run them through a program called Foliage that calculates the percent ground cover in each photo. For example, the photo shown here has 68.4% ground cover. We then take the percent cover number and multiply it by the average plant height in that field to get an estimate of cover crop biomass. So we just wrapped up our first season of data collection this past spring, where we were able to gather data from 129 cover crop fields across Michigan, Ohio, Indiana, Illinois, Wisconsin, and Minnesota. 62 of the fields were single species cover crops and 65 were mixtures. And we had significantly higher levels of participation in Michigan and Wisconsin. So this coming spring, we're really hoping to achieve better representation across all 6 states. In terms of results, we found that biomass for both single species and mixture cover crops varied really widely across the region, with single species biomass shown on the left here and mixtures on the right. However, when we compare the 2 graphs against each other, we see that the single species cover crops actually showed a much larger range of variation than the mixtures, from a minimum of only 0.5 pound per acre to a max of over 7,200 pounds per acre, whereas the mixtures had a minimum of 42 pounds per acre and a max of over 4,300. So this trend is reflected in the coefficients of variation, or CV, where even after controlling for growing degree days, the single species fields have a CV of 1.25, while the mixtures have a CV of 0.96, which tells us that overall, the single species fields are quite a bit more variable than the mixtures. Another interesting thing to note here is that despite the maximum biomass value being quite a bit higher for single species covers than for mixtures, The mixtures do still come out on top when we look at average cover crop biomass, which was 983 pounds per acre for mixtures compared to the single species average of 892. So all of this taken together suggests that mixtures may be more reliable than single species cover crops, potentially because having multiple species growing does indeed help increase the chances of at least one species doing well. However, this still doesn't solve the problem that in general, across all of these fields, both types of cover crops are still really variable. So my next few slides address the question, what is driving all of this variation? So thinking big picture at the regional scale, one of the things we might expect to have an important influence on cover crop growth is climate, right? Which is indeed what we found. So these graphs show that rye biomass on the y-axis increased with growing degree days shown on the left, as well as with precipitation, which is shown on the right. However, again, there's still quite a bit of variation here left unexplained, which is where variables from the Farm and Management Survey that participants completed come into play. So these three graphs show relationships between rye biomass and three different management variables: planting date in the upper left, seeding rate in the upper right, and then tillage intensity on the bottom. As expected, we found that later planting dates in the fall are associated with lower rye biomass in the spring, although it is still quite impressive that some growers were able to get rye planted and established as late as November last year. We also found a positive relationship between seeding rate and rye biomass, although it's somewhat weak compared to the other relationships we've seen so far, suggesting that seeding rate may play a relatively minor role, at least at the regional scale, in terms of influencing cover crop growth. And then the bottom figure shows the relationship between tillage intensity and rye biomass. The tillage scores were calculated based on tillage practices from the past 5 years, as well as historical tillage use on each field, where higher scores indicate higher levels of soil disturbance over time. So I initially expected to see a negative relationship here, since greater soil disturbance can negatively influence soil health. But instead we found this positive relationship, which could be due to the fact that tillage helps improve seed soil contact for rye. For cover crop mixtures, one of the interesting things we've found so far is that mixture composition does seem to really matter in terms of predicting overall mixture biomass. So the left graph shows that the number of different species included in the mixture, also known as species richness, had no influence on overall mixture biomass, whereas the graph on the right shows that mixture functional diversity, or the number of different types of species, was positively associated with mixture biomass. So this suggests that simply adding more species to a mixture isn't always beneficial for overall biomass, and that instead there may be greater benefits to sort of strategically combining species with different traits and characteristics. And then this slide shows comparisons between how cereal rye biomass and mixture biomass respond to different planting and nutrient management strategies. So the two graphs on the left, with cereal rye on the top and mixtures on the bottom, shows that cereal rye may benefit from being drilled compared to broadcast or aerial seeded, whereas mixture biomass doesn't seem to be strongly influenced by planting strategy, which I think makes sense for mixtures since they often contain seeds of different sizes, which can make it challenging to get drill settings calibrated just right for each species. And then on the other hand, the fact that rye does do better when it's drilled, again, suggests potentially an important role for seed soil contact with rye. And then in the two nutrient management graphs on the right, we see similar trends for both rye, again shown on the top, and for mixtures, which are shown on the bottom. So there currently doesn't seem to be any major difference in biomass between cover crops that did receive fertility amendments like manure, fertilizer, compost, and those that didn't. So this indicates that applying nutrient amendments directly to cover crops may not actually be worth the extra time and cost. So keep in mind all of these results I've just shared are still preliminary, and we're really excited to be moving forward with a second season of data collection this coming spring. So certainly stay tuned for more results next year. All right, so now we're going to move on to the on-farm experiment that I mentioned, where our goal is to understand how soil health specifically influences cover crop performance. So the results I'm sharing for this are part of a 2-year study that my advisor, Jennifer Blesch, is leading on 15 farms across 2 different regions of Michigan. But today I'll just be focusing on work that I did on 6 of those farms located in the Thumb region of Michigan during the first year of the study, specifically looking at cover crop roots. So in August of 2020, we planted 3 cover crop treatments in each field, including a cereal rye monoculture, which is an overwintering grass, a crimson clover monoculture, which is an overwintering legume, and a functionally diverse mixture of rye, clover, dwarf Essex rapeseed, which is an overwintering brassica, and oats, which are a winter kill grass. So although all the fields in this study are now managed organically, they have really diverse management histories, ranging from several decades of organic practices to those just recently beginning the transition to organic management, which of course can result in really pretty large differences in soil health across farms. So we characterize those differences using a suite of biological, physical, and chemical indicators listed in the table on the right. We then returned to each of the farms in the spring to collect root cores from each of the 3 cover crop treatments and separated the roots into 3 different size fractions, so fine, medium, and coarse roots based on root diameter. And the reason we did this is because those 3 different root fractions serve different functions. So fine roots are really important for soil resource uptake, while the medium roots are really important for helping to transport those resources throughout the plant. And then the coarse roots are the big tap roots that you often see for brassicas that can help with reducing soil compaction. And in this particular study, the dwarf Essex rapeseed was the only species that fell into the coarse root category, while the rye and clover were the 2 species making up the medium and fine root categories. We then also measured different plant traits or properties of those roots, including root carbon to nitrogen ratio, root to shoot ratio, which is the ratio of above ground to below ground plant material, and fine root biomass and nitrogen concentration. And then lastly, we also measured several ecosystem services provided by those roots, including total below ground biomass, nitrogen assimilation by those roots, as well as carbon inputs to soil from the standing root biomass. And we found that on average across the 6 farms, the functionally diverse mixture outperformed the rye and clover monoculture treatments in terms of total below ground biomass, nitrogen assimilation, and carbon inputs. So this was largely due to the coarse roots of rapeseed in the mixture, which had really thick, deep tap roots that complemented the dense fibrous roots of rye and the smaller tap roots of clover. So overall, this result really underscores the importance of below ground functional diversity for enhancing mixture performance compared to single species cover crops. When looking at carbon to nitrogen ratios, which is of course an important indicator of residue quality and decomposition, we see that the coarse roots had a really large impact on the average C to N ratio in the mixture, such that the mixture actually had the highest overall C to N at 35.2, followed by rye and then by clover. However, we do also see that the medium roots in mixture do show an intermediate C to N between the rye and clover treatments. So it is possible that by combining species with contrasting C to N ratios in mixtures like we see here, could potentially lead to improved synchrony between cover crop nutrient release and nutrient demand by the subsequent cash crops. And then for root to shoot ratios, we saw that it was significantly different across all three treatments, with rye having the highest root to shoot ratio, which is what we would expect given its really expansive root system. Clover had the lowest root to shoot ratio, and then the mixture had an intermediate ratio between rye and clover. And I think the fact here that both the rye and mixture treatments had root to shoot ratios above 1 really highlights the large contribution of roots to total cover crop biomass. So I think roots are certainly an important part of the picture that should be given some more attention. So despite all of these strong overall trends across the three treatments, we also found significant differences in cover crop outcomes within each treatment across the 6 farms. So these figures again show below ground biomass, nitrogen assimilation, and carbon inputs for each treatment, but now broken down by site to emphasize that the performance of each treatment is indeed context dependent. Another interesting thing to note is that although the coarse roots in mixture were highly variable, the combined medium and fine root fractions were the least variable in the mixture. For example, with a CV of 0.24 for below ground biomass compared to 0.29 and 0.38 for clover and rye, respectively. So this is likely because the functional complementarity between rye and clover and mixture allowed for more stable performance across sites than when either species was grown alone. So in other words, in fields where the rye didn't do well, the clover tended to make up for it, and vice versa. But again, this begs the question, what is it that's driving variation across all of these species on these farms? So if you'll recall from the beginning of my presentation, I gave you a sneak peek and said that part of the reason these two rye cover crops turned out so different was due to the soil. So these photos are indeed from plots in this on-farm experiment. And the next few slides are going to showcase exactly what it was about the soil that ended up predicting differences not only in rye performance, but also for clover and rapeseed across farms, still focusing on the cover crop roots. So I know this table has a lot going on, but I promise I'll walk you through all of it. It shows correlation coefficients between soil health parameters and rye root properties. So basically showing how related or unrelated different soil properties are to the root outcomes that we observed in the field. And so far, we've found 3 really important relationships for the first year of data. First, we found that potentially mineralizable nitrogen, which is an indicator of soil biological activity and internal nutrient cycling capacity, was negatively related to rye fine root biomass, suggesting that rye didn't invest as much energy below ground to scavenge for nutrients in soils where nitrogen was readily available. And second, there was also a positive relationship between soil phosphorus and rye fine root nitrogen concentration, which aligns with previous findings in our lab that show higher root nitrogen concentration when other growth-limiting nutrients like phosphorus are plentiful in the soil. And lastly, we also found a positive relationship between soil phosphorus and rye root to shoot ratio. So we expected rye root to shoot to decrease with higher levels of nutrient availability, since the roots don't need to proliferate as much in cases where nutrients are already readily available in the soil. But this positive relationship could be driven by the fact that soil phosphorus was strongly related to clay content. So farms with higher clay content and phosphorus content also tended to have lower levels of soil biological activity, which could be because they had historically relied on soil texture to provide fertility, and thus were later adopters of soil health practices like cover cropping compared to other farms in the study. And then for clover, we again found that potentially mineralizable nitrogen, that important predictor of soil biological activity, was again an important predictor of several root properties here. Specifically, clover below ground biomass, nitrogen assimilation, carbon inputs, and fine root biomass all increased as mineralizable nitrogen decreased, which makes sense given that clover is a nitrogen fixing plant, which means that it tends to get sort of lazy and have lower growth in soils where there's already a lot of nitrogen available. We also found that several clover root properties were negatively related to soil clay content, which I believe is actually due to the fact that the clover roots ended up having a difficult time penetrating the denser soils. And then lastly here, we also have a negative relationship between soil phosphorus and several clover root properties, which was somewhat surprising at first because legumes are often phosphorus limited. But I think these negative relationships may actually be reflective of the fact that the soil phosphorus availability is strongly related to clay content, as I mentioned in the last slide. So in places where we see strong relationships with clay, they also tend to be reflected in relationships with soil phosphorus. And finally, for the brassica in mixture, rapeseed, we found that the single most important soil predictor, again, was mineralizable nitrogen, with a clear trend of greater below ground productivity and higher root nitrogen concentration with increasing mineralizable nitrogen. And then compared to rye and clover, the rapeseed roots seem to be much less responsive to soil phosphorus. So just to tie everything together here, I know that was sort of a whirlwind of information. What I'm hoping you'll take away from this presentation is that cover crop performance, both above and below ground, can vary in response to a wide range of environmental and management factors. And this means that it's really important that we devote more time and resources to research on real farms that focuses on identifying the most important factors influencing cover crop outcomes in order to better create context-specific recommendations for cover crop management across these different growing conditions. One of the most promising things I found in my research so far is that functionally diverse cover crop mixtures can be more reliable and often outperform single species cover crops across a wide range of conditions. So if you're looking for one piece of management advice to take away from this, I think it would be to start learning about and experimenting with cover crop mixtures if you haven't already. And finally, we also have this wonderful growing body of evidence, including what I presented here, suggesting that biological indicators of soil health, so how well the microbial communities are functioning in your soil, like potentially mineralizable nitrogen, are really important predictors of cover crop outcomes across farms. So we have more research on the way that continues addressing these themes in order to help improve agricultural and environmental sustainability across farms. And with that, I'm happy to turn the floor over to questions, and I hope that this has hopefully been interesting and informative.
Thanks, Etienne. And before we get to questions, and there are many questions, I need to share my screen quickly. Yep. For two items here. First, hopefully you can see this. If you watch this webinar and you want to earn a CCA CEU, please send your name, the name you entered to watch the webinar, and your CCA number to Elena Whitaker, and her email is ALENAW, Elena W @iastate.edu by 5 o'clock today. And then I want to put a plug in for the first webinar of 2023, which will be Dr. Keith Schilling, that's the state geologist and director of the Iowa Geological Survey at the University of Iowa. Dr. Schilling um, is going to talk about floodplains as conservation opportunities. So please tune in January 4th for Dr. Schilling. And again, if you need a CCA CEU, please get your name, the name you entered to watch the webinar, and your CCA number to Elena W at iastate.edu. Now, Etienne, I will let you share your screen again, and we will go to some of the questions. So one of the questions was about kind of how the landowners were chosen, the participants chosen, and are you looking for additional farmer participants in the future?
Yeah, I love that question. So it was completely voluntary. I basically reached out to as many different people as I could about the study, and folks who were currently growing cover crops. Anyone who was growing a cover crop was eligible to participate. So we are certainly recruiting more farmers to participate for this coming spring. And if you're interested or know people who might be interested in participating, please do shoot me an email. And I would be happy to share the sign up information with you and answer any questions you might have. You can also visit the Blesh Lab website, and we have some more information on there as well.
And one connection into the chat box that you might be able to get. Okay. What was the name of the software you used to determine the percent ground cover with the different cover crops?
Yeah, so it was called Foliage. And some of you may be familiar with an app called Canopio, which does the same thing. But Foliage is specifically for like batch processing. So I had like hundreds of photos to process, and it just does it much more quickly than Canopio does. But if you're interested on your own property to look at some of those things, you can certainly download the Canopio app on your phone and take some photos, and it'll calculate that for you.
Great. How was the shoot and root biomass collected and measured? And did the farmer participants collect these samples?
Great question. So when we were looking at the cover crop roots, it was just the research team that went out and collected the samples. And so we basically had these, they're called Dutch augers, but they're basically just these large tubes, cylinders that you insert into the soil. And then you might have a picture of it here somewhere. I don't think I do, but you then remove the auger from the soil, and we just collect the soil into plastic bags and then take it back to the lab. And we go through this really extensive washing process where you rinse the roots off, you clean all the soil off, you use sieves of different sizes to isolate the different root fractions. So it's a pretty involved process, but we're excited with the data that we have so far from it.
Yeah, anytime it's an important indicator, but not the easiest to measure, the root biomass.
Is challenging, yes.
Yeah. Another one is, why do you think landowner engagement and participation was lower in some states compared to Michigan and Wisconsin?
So in Michigan, I think it was in large part due to the fact that I just have way more connections here. Like I live in Michigan, my university is in the state. And then in the case of Wisconsin, I just ended up being really lucky to connect with a woman who was involved with these farmer-led watershed groups who were all of these farmers were using cover crops and were super excited about participating in research. So that ended up, I think, being the main reason why we had so much participation in Wisconsin, but really trying hard this year to get more folks from the other states too.
Okay. And this one, I think you answered, but what was considered in the below ground biomass? Any microbes or just plant biomass?
So we didn't look at like specific microbe species per se, but we did look at things like the activity of the microbial communities and things like that. And then that's part of what we're using to understand root outcomes on these different farms, like linking the activity of those microbial communities to, whether or not root biomass is higher on one farm versus the other. But there's also a postdoc in the lab that I'm in who does do much more detailed work looking at the composition of microbial communities. And so he'll hopefully have some papers coming out soon with more detailed information on that.
Okay, great. And did you collect any information on why they might have used single species cover crops versus mixtures?
I don't think we did. That's a really good point. We did collect information on like the number of years that folks had been using cover crops. And I think a lot of folks who were newer to using cover crops sort of we're more likely to use cereal rye just because it does tend to be pretty hardy and reliable. And then folks with more experience, I think, also tended to be the ones who were experimenting with these crazy mixtures with like 14 different species, just because they had more experience. And I think we're excited to start like pushing the boundaries. But yeah, that's a great idea. I could potentially add a specific question asking why they chose the specific cover crop that they did.
Were any of the sites grazed?
Yes. So in the citizen science study, we had a question asking if the cover crops had been grazed, like intentionally or unintentionally. So we do take that into account when we're looking at differences in biomass.
There was a question here, maybe just describing a little bit in more detail what you mean by the functional winter diversity in one of the kind of earlier slides in your presentation.
Absolutely. Yeah, let me go back to that slide. I think perhaps this one. So yeah, functional diversity is sort of this complicated topic because you can measure it in different ways, you can define it in sort of different ways. But basically, it just gets at this idea of species having unique characteristics. So you could think about, like in addition to the examples I have here, where these species, like one of them can basically create its own nitrogen. The other one like scavenges soil nitrogen really well. So they're using like nutrients in different ways. But you could also think about like the phenology of species. So basically, you could combine a species that grows really quickly in the fall but dies off once the winter hits, it frost kills. You could combine that with a species that overwinters or is more frost tolerant, and that could be really beneficial too because you're sort of like distributing the plant growth potential across a longer duration. So there are a lot of different ways. You could also think about like shade loving versus sun loving species, basically just combining species that do different things in your mixtures. I hope that helps.
Yeah. Another one here. Do you know if farmer experience with cover crops play a role? For example, or first year use versus several year use?
Mm-hmm. Yeah, so far from the first year of this study, there doesn't seem to be any relationship between the level of experience that a farmer has and the biomass outcomes. So it's not like we see that farmers with more experience have higher cover crop biomass compared to the ones who are using cover crops for the first time, which I think is a really interesting outcome. But I would also really love to increase our sample size this coming year and see if perhaps we do start to see some trends emerging there. We are particularly interested in adding more folks who have used cover crops for like 20 plus years and like first year users. We have a lot of people between like 2 to 15 years. Yeah.
Great. Okay. There was one. Did you measure soil compaction?
So in that on-farm experiment, we did measure soil compaction. It's called bulk density. And so we just take these cores from the soil. We know what the volume of the core is, and then you can basically it's the weight divided by volume, and you can look at how compaction varies across the different fields.
And did you collect information on how the cover crops were terminated?
Let's see. I don't, so in the citizen science study, we don't have data tracking how they were terminated, unfortunately. The only thing we asked about was sort of like the target termination date. Yep. So we do have that.
And I'm assuming with the farmer citizen science, they were using their own equipment or whoever they had for they weren't necessarily supported by outside resources to do their cover crops necessarily as part of this study, at least.
Exactly. Yep.
Okay.
So the study was basically just like totally observational. So we collected information on what the farmers would be doing regularly, like if we weren't, imposing any research limitations or anything like that.
Um, do you know if you have any of those sites utilizing interseeding early, such as in the June or July timeframe for interseeding the cover crops?
We did have a few of those, yes. So I'm hoping to sort of dig into the data in a bit more detail to see how that might play a role, but we did have a few folks who were doing that.
Great. I guess that there was a question, how and when were the on-farm demonstrations seeded, but it would be kind of based on whatever those individual landowners were doing in that situation.
Yeah, so for that first study, the citizen science, it was totally up to the farmer, but for the on-farm experiment on that small subset,
Yeah.
We did standardize the way that the cover crops were planted. They were all following a small grain. And then the fields were tilled prior to cover crop planting, and they were all three treatments were drilled. Yep.
Okay. And I guess that makes me think of one. When you look at the growth of the single species versus the mixtures, did you break out what crop it was following?
In the citizen science, yes, we did.
Okay. Okay.
I do have data on that if folks are interested, but could only squeeze so much in here.
Okay. Well, when you said small grain, planted after small grains, I was thinking that, a lot of our areas, we don't, I know in Michigan and parts of Ohio, you have more small grains than we do in Iowa, with just almost everything's coming after a corn or soybean crop.
Right.
Yeah.
Yeah, I think one really interesting tidbit related to that so far, I did actually see quite a few fields that were coming out of corn or soy where they did manage to plant mixtures. So that's one thing I'm hoping to follow up on to figure out like what the mixtures were composed of, how they planted them, that kind of thing, because it would be lovely to see more folks using mixtures after corn and soy.
Yeah, and see how much growth, because I know in some studies we've done in Iowa, it's been a little challenging to get a lot of the mixture growth just after corn or beans.
Right.
So. Were the participants always focused on achieving higher amounts of cover crop biomass, and is there any risk associated with too much biomass?
Yeah, that's a good question. So for the citizen science stuff, like we sort of imposed that goal onto the cover crops. Like we just assumed because so many ecosystem services are directly related to the amount of biomass that accumulates, we use biomass sort of as a proxy for like overall performance. But one thing I'm really interested in studying in future work is sort of how farmers define performance, right? It could be measured in a lot of different ways. So I think that's a great question. But in that on-farm experiment, we actually did measure all of the different ecosystem services. So like we measured weed suppression, we measured like the nitrogen supply from legumes, nitrogen retention from the soil, like the recycling of nutrients component. So that one is much more multifaceted, if you will. We looked at much more than just biomass.
Next one is, does the percent composition matter for diversity, or do you just have to have it? Kind of somebody's thinking about keeping those costs down.
Mm-hmm. Yeah, also a good question. And I think that's like a very active ongoing area of research because of course, like legumes tend to be more expensive than grasses when you're planting a cover crop. So like, what is that threshold level of legume biomass that you need in order to get the benefits from them? So that's certainly something we're interested in understanding and will be part of that aspect of sort of like creating these context specific recommendations. Like if you have poor soil health, is it better to increase the proportion of legumes compared to grasses and vice versa? So that's something we're looking at.
I think that really gets most of. There was a question. They may have. I think you talked about this, but did all the participants use no-till, or it sounded like there maybe was some tillage associated with that citizen science study?
Yep. Yeah, we did have a really high proportion of folks using no-till, but we did still have folks using conventional tillage as well as a number of people using like reduced tillage, which I think I need to be a little bit more specific about this coming year because reduced tillage could mean a lot of different things to different people.
Different.
But that was sort of how we categorized it, like no-till, reduced till, or conventional.
Okay. Okay. And maybe you could bring up the slide of the soil health parameters that you collected in that more detailed study.
Yep.
And I guess, they wondered if the data was available. My impression was this hasn't been published yet.
It has not been published, but I do at the end here have sort of like a summary
Okay.
table for the
Okay.
field sites. So
Okay.
yeah, we did see quite a bit of variation in terms of those biological parameters, as well as texture. So yes, keep an eye out in the
Okay.
next year or so for official papers with all of this data.
All right. So again, if you're on, I posted in the chat box to everyone to see if you need a CCA credit, email Elena w@istate.edu by 5 o'clock today with your name, the CCA number, and the name you used in logging into the webinar. And then again, next month in 2023, January 4th, we'll have Keith Schilling talking about floodplains. This person was looking if you had done any infiltration measurements on any of the sites.
No, that's a good one, but we haven't actually incorporated any of that so far, but
Okay.
potentially for the future.
Okay. Yep. Even more field work. That's I don't know which is worth, root biomass or infiltration. Either one of them are
Right.
pretty challenging. So, Etienne, thank you so much for your presentation. It was an excellent way to end 2022 with a great webinar on cover crop. So I just really want to thank you for your presentation and participating in our Iowa Learning Farms webinar. And hopefully, when you get even more results, you might be willing to come back and talk to us again. So with that, I don't know if you have any last comments.
No, yeah, thank you so much for the opportunity. It's been great.
Okay, with that, I will stop the recording.